It's a great pleasure to talk about LifeArc's assets in the area of Alzheimer's disease and the progress we made during the last 14 years, 13 years or something like this. Okay. Actually, this is all we need to know. The amyloid cascade nowadays, it's not longer a hypothesis, it's proven, it's clinically clear and proven. This is the trigger for Alzheimer's disease for sporadic and also for the familiar forms. But what is also proven is that the Alzheimer plaques and the physiological peptide, the A beta 1 to 40, 1 to 42, which we produce all of us. As we're sitting here, we're producing these peptides. And of course, we are not demented. We are fresh and clear with the coffee and everything is fine. So why should we remove it? So that's the major idea. Why should we remove these physiological peptides, although we know they can accumulate and build up these plaques? So that was the original idea quite many years ago, that the plaques are to be removed from the brain and the patients are being healed. This was overall the main idea, but it's proven wrong. And it's also proven wrong that these physiological peptides, these A beta 1 to 40, 1 to 42, should be removed. All efforts in clinical trials, phase 3, failed, actually. So, and quite a while ago, we had the idea, maybe we can find an antibody which does not react with the physiological peptide, and it should not react with the plaques. But nowadays we know that besides the full-length peptides, there are N-truncated, a little bit short peptides at the N-terminus, either the 3 amino acids away, so A beta 4 to 42 or pyroglu 3 to 42. Maybe we can find antibody recognizing these two peptides, but not the physiological and not the plaques. So that was the starting point. And actually we did. The type 1 antibody, this is a staining scheme in Alzheimer's disease brain, where the type 1 antibody just sees a little bit plaques, but no comparison to humanized antibody staining, babineuzumab with Alzheimer's disease brain or desolanizumab. So just a little bit plaques. And we then beefed up with LifeArc, and LifeArc then humanized the type 1 antibody and identified more than 50 variants. And we also, all the time, we wanted to make sure that the binding properties of the original murine antibodies from the mouse kept the same features during the humanization process. And the type 1 antibody should bind the N-truncated A beta 40 42, it should bind the pyroglu 3 to 42, but it should not bind the physiological peptide, and it does not. And all of the lead candidates, type 101, type 102, type 103, and now the best one, type 104, they all had the same features. They did not bind to the physiological peptide, but to the pathological peptides. And moreover, is there one lacking? No. Okay. And moreover, in a crystallization effort together with the University of Leicester, LifeArc then crystallized the type 1 antibody with the N-terminus of the pyroglu peptide. And by doing so, they identified a very clear and novel structure which has never been studied before and never been detected before. The peptide makes a so-called pseudo-beta hairpin conformation, so it's bending back during the binding in the antibody binding pocket. And this now is stable, but of course when the peptide is released from the binding pocket, it's no longer stable and it's soluble. But then they came up with the idea, well, maybe we can stabilize this structure by adding a covalent bridge within the A beta 1 to 14 peptide, and it worked. So this now is the cyclist A beta 1 to 14 peptide, which has the same structure as the bound one with the antibody. And now we have a vaccine. But before we proceed to therapeutic efforts, we had to be sure that our antibody, the type 1, is the only one binding to this new structure and not any of the competitor antibodies. And this is now a binding experiment where you can clearly see only type 1 can bind the cyclist A-beta 1 to 14, but not of the other humanized antibodies, even those antibodies which have been directed against the pyroglutamate. So it's completely different structure, completely new structure. But is it therapeutically evident, and has it the same features as the humanized antibody? So it should not recognize plaques, of course. Although it's an antiserum which has been injected in rabbits and mice, and then we looked if the binding properties are still the same, and they are. Here you see Alzheimer's disease brains stained with a typical plaque antibody like the solanezumab or research antibody, the type 104. This is the lead candidate, humanized one. See just a little bit staining, and even the antiserum obtained after this immunization with the cyclist A-beta 1 to 14 shows very, very minor plaque staining. So this was very promising. Then we wanted to know, are these, the antibody and the active immunization protocol, are they therapeutically evident? So we made a series of experiments. I only want to show you some of these experiments. This is the brain scan of an typical Alzheimer mouse. Here is the brain scan where we visualize the consumption of glucose uptake. And of course, a wild-type mouse, you can see that it's brightly enlightened. There's a lot of glucose uptake. Of course, wild-type mouse is a normal brain. However, the Alzheimer mouse, the name is 5XFAD, the 5XFAD mouse has very, very low glucose uptake, as it should be. So this is the controlled passive immunization. When we made a passive immunization with the type 104, the glucose uptake is a little bit higher. The control with the active immunization, of course, like the other control, and the active immunization now with the cyclist A-beta 1 to 14 clearly shows an elevation of the glucose uptake, and this is, of course, statistically significant. Next, we wanted to know, is this treatment on reducing the plaque load? Although we know neither the antibody nor the antiserum against the cyclist A-beta 1 to 14 is reacting with those antibodies. So we know the epitope is a soluble oligomer and not the plaques. So these mice here are ideally suited to study the plaque load in a mouse model. And of course, again, wild-type mice, they have no plaques, so there is no retention signal. And an untreated Alzheimer mouse has a lot of plaques. Of course, we do see a lot of the material highlighted. After passive immunization with the lead candidate, the plaques are reduced. This is the control. Active immunization, of course, no effect on the plaque load. And the plaques are virtually gone after the active immunization with the cyclist A-beta 1 to 14 peptide. So, and this is also verified by staining of the brains, of the brain sections. So after passive immunization or active immunization, the plaque load is clearly reduced in all instances. So showing that not only the brain scans are showing this treatment effect, but also on the brain section level. Next, but this is even more important because many humanized antibodies have been shown that they're having effect in mouse models on the plaque load. So what? We know that the plaques per se are not so important, And if this is the only measure if you take, maybe this is not enough. So we developed an own mouse model. Here in this case, this mouse model expresses only the A-beta, the human A-beta 4042 peptide in the hippocampus, in the pyramidal neurons of the hippocampus mainly. And these mice develop a severe neuron loss after months. So after 5 months, 50% of the neurons are gone. And we wanted to know what happens if we use that mouse model with such a robust pathology on the neuron level when we use the Tabo 104 for passive immunization or the immunization protocol. And here you can see a neuron count, just counting the neurons in the hippocampus. Here the CA123 region after these treatment approaches. And you can clearly see that both the passive immunization as well as the active immunization leads to more neurons where you want to have it. So this is really interesting because both the passive and the active immunization is done in the periphery. So the antibodies are circulating in the blood, of course, in both treatment schemes, and neurons are gone in the brain. So that also shows us that we can reach targets within neurons. Nobody knows how, but I mean this is the experiment for it. So we are not asking why this is, but it's clear that we also can rescue the neuron loss in such a robust mouse model. And the last experiment then with this mouse model, this is a so-called Morris-Mortimer test. You can ask the mice to show if they can have a reference memory, sort of a cognitive test for mice, where they learn to find a hidden platform. So the mouse is circling in the basin with the opaque water. Only we can see the platform. The mouse cannot see it, but has to learn it. And all the mice usually, they learn it perfectly. This is no problem. And then we place the mouse at a certain location. The mice are traveling and locating at different cues, and they know, oh, okay, at platform, at a certain cue, there is the platform, and they can get off. However, Alzheimer mouse models cannot do that anymore, at least this TG442, because the neurons are lost, and if you don't have the neurons, of course, they cannot navigate and find a platform. That's clear. So the target quadrant is the one quadrant where the hidden platform is. A little bit complicated, but they need to do that pattern. So they had to circle in the target quadrant. So to show us, okay, they had learned the task. And only after the passive immunization or after the active immunization, the mice performed the same way as wild-type mice. So that proved again on that level, not only the neurons are rescued, but also the cognitive behavior of the mice is rescued. So, the LifeArc therapeutics are really uniquely positioned in the area of Alzheimer's disease. I mean, usually the A-beta antibody biology in Alzheimer's disease is clearly crowded, but the unique structure clearly drives an excellent IP positioning, because this is the only antibody where a clear structure has been shown. All the other antibodies have a binding to plaques, to beta structures, to beta sheets, but this is not defined really. Here in this case, LifeArc has a very clear epitope, and this epitope is completely novel. Moreover, we can also say that the selectivity and the safety is also very clear, because the selectivity targets only the end-truncated peptides, and these are those peptides which come later in the Alzheimer pathology. And we omit the normal physiological peptide A-beta 1-40, A-beta 1-42, what we all produce as we're sitting here. And also, because we avoid binding to plaques, we will not target them in the first indication, and this plaques are sort of waste bins. Waste bins full with toxic material. Why should we touch it? If they are there, this is a safe place for this toxic material. And our antibody and the vaccine do not touch the plaques. So this is also much safer than those antibodies produced by the competitors, which should target the plaques, what they do. It's highly efficacious because of the same reason, because if this little antibody fused into the brain through the blood-brain barrier is soluble in the brain and do not find the plaques, which they don't, it's clearly efficacious. Then we have also shown preclinical success in many in vitro and many in vivo situations. So finally, that's the end, and I want to thank all the people who contributed to the work. Happy to take questions.
Thanks, Thomas. Just in a super exciting program that we're working with Thomas to bring to the next step of translation. We have time for a few questions.
Over there.
Hello. Thanks very much. Very clear talk. So one of the things you were telling us is that the amyloid is being deposited into plaques over many years. So I wondered when, in the course of either pre-disease or once a kind of mild cognitive impairment or frank disease, when do you think your two therapies, because you've got the antibody, active and passive, would need to be delivered to patients?
Well, the antibody cannot be delivered much prior to the disease because of the cost, and you have to have a diagnosis for Alzheimer's disease. That's very clear. This is comparable to the other antibodies, but the situation is completely different for the vaccine. This can be given in the population prior to disease, because this is safe and it cost effective. That's what I'm thinking. If the antibody is working, the vaccine will be much better situated on the market, I think.
Paul Morgan, UKDRI Cardiff. That's fantastic stuff. Really, really interesting. Just first on the point you just made about early diagnosis, early treatment being so key here. I mean, that's an issue still with Alzheimer's disease, is we just don't have the tests yet that enable us to do that. And so that could be a problem. I'd like to know whether you think your agent will actually work better in later disease where we can actually make the diagnosis and treat now. And second, you said the plaques are kind of inert. They're just dustbins full of rubbish and you just can leave them alone, nothing much happens. And I absolutely agree with you, but I don't think the evidence actually is fully accepted. The plaques themselves are not driving the disease process. Can you comment on that?
Absolutely right. This has been a conflict for many years, for the second question. For many years because only the tau pathology is sort of aligned with the clinical symptomatology. Absolutely right. And the plaques are just everywhere. They appear, I think, 20 years before the onset of the symptoms and in the frontal cortex, in temporal cortex, but not where the clinical symptoms are failing. So, therefore, there was really a battle between those saying tau is to be targeted first, and those against A-beta should be targeted first. Now we have the clinical trials. And the clinical trials clearly say, I mean, from donanemab or Leqembi, for instance, clearly saying if you target A-beta peptides, you get also a reduction of the tau pathology. So I think the amyloid pathology, how it does work, nobody knows, but the amyloid pathology is clearly first that triggers the tau pathology. I think that can be said. And then the testing, that's really crucial. But as far as I understand, the field, blood testing for tau, phospho-tau, is quite advanced. Even on a routine basis, but now I think only in selected populations it's really working. But the question is, in the clinical, and a routine is not established now, but I hope this will be coming.